Most of oxygen in blood is present bound to hemoglobin molecules in red blood cells, and around 1 to 2% is present in dissolved form. Oxygen from dissolved form can bind to hemoglobin, and oxygen from hemoglobin can dissociate and become part of dissolved form. So this reaction can occur in both directions. Later on, we'll discuss factors that affect direction of this reaction. Oxygen bound to hemoglobin is measured as percent saturation of hemoglobin or simply oxygen saturation. And oxygen present in dissolved form exerts a partial pressure and is measured as partial pressure of oxygen or PO2. Now let's discuss factors that affect binding or dissociation of oxygen from hemoglobin. These factors affect uptake of oxygen in lungs and delivery of oxygen in tissues. One of the main factors that affects oxygen binding and dissociation is partial pressure of oxygen. Partial pressure of oxygen affects binding or dissociation of oxygen even if the affinity of hemoglobin remains same. Suppose we have a hemoglobin that has initially low saturation and affinity of hemoglobin remains same, and we expose this hemoglobin to different partial pressures of oxygen. If partial pressure of oxygen is high, oxygen will bind to hemoglobin and it will gradually increase oxygen saturation of hemoglobin. And if partial pressure of oxygen is sufficiently high, saturation of hemoglobin will keep increasing until hemoglobin becomes fully saturated with oxygen, and at that point saturation of hemoglobin will be 100%. On the other hand, if partial pressure of oxygen is low, oxygen will not bind to hemoglobin and saturation of hemoglobin will not increase. And if oxygen saturation is initially high and partial pressure is low, oxygen will dissociate from hemoglobin and it will increase the amount of dissolved form and it will increase the partial pressure of oxygen. So partial pressure of oxygen is one factor that affects binding or dissociation of oxygen from hemoglobin. And in lungs, partial pressure of oxygen is high that increases oxygen saturation of hemoglobin, and in peripheral tissues, partial pressure of oxygen is low that helps in dissociation of oxygen from hemoglobin and helps in delivery of oxygen to tissues. Another important factor that affects oxygen binding or dissociation from hemoglobin is affinity of hemoglobin for oxygen. Affinity of hemoglobin for oxygen affects oxygen binding or dissociation even if the partial pressure remains same. Affinity means how readily or easily oxygen binds to or dissociates from hemoglobin. Suppose we have hemoglobin that initially has high oxygen saturation and partial pressure of oxygen in solution is low. So if hemoglobin has low affinity for oxygen, oxygen will readily dissociate from hemoglobin and it will increase the amount of oxygen in dissolved form. In tissues, this will help in delivery of oxygen to tissues. So in tissues, we want hemoglobin to have low affinity for oxygen. On the other hand, if affinity of hemoglobin for oxygen is high, oxygen will not readily dissociate from hemoglobin and less amount of oxygen will be released in solution. So in tissues, we do not want hemoglobin to have high affinity for oxygen. On the other hand, if partial pressure of oxygen is initially high and oxygen saturation of hemoglobin is low, then if affinity of hemoglobin for oxygen is low, oxygen will not readily bind to hemoglobin and oxygen saturation of hemoglobin will remain low despite high partial pressure of oxygen. And if partial pressure of oxygen is high and affinity of hemoglobin for oxygen binding is high, oxygen will readily bind to hemoglobin and it will increase saturation of hemoglobin. So in lungs, we want hemoglobin to have high affinity for oxygen. So oxygen can readily bind to hemoglobin. So we want hemoglobin to have high affinity for oxygen in lungs and low affinity for oxygen in peripheral tissues. In fact, in body, hemoglobin has high affinity for oxygen in lungs and low affinity in peripheral tissues. So how it is possible that hemoglobin has high affinity for oxygen in lungs and low affinity in tissues? To understand that, we need to understand structure of hemoglobin molecule. Each hemoglobin molecule consists of 4 globin chains, and each globin chain has a heme molecule, and in center of each heme molecule iron atom is present. Oxygen actually binds to this iron atom present in center of heme molecule. So each hemoglobin molecule can bind up to 4 oxygen molecules. Hemoglobin molecules exist in two forms. When no oxygen is bound to hemoglobin, iron atoms in hemoglobin are not readily accessible for oxygen binding. So affinity of hemoglobin for oxygen is low when no oxygen is bound to hemoglobin. This state of hemoglobin is called 10 state or T state, and this state is maintained due to stronger ionic interaction among adjacent globin chains. So when hemoglobin is in tense state or T state, it has low affinity for oxygen binding, and this state is favored in peripheral tissues, so this helps in dissociation of oxygen from hemoglobin. However, when first oxygen molecule binds to hemoglobin, it results in some conformational changes in hemoglobin molecule. And these changes are due to changes in ionic interactions between adjacent globin chains. And due to these changes, there is some rotation of adjacent globin chains. And this rotation results in remaining iron atoms more readily accessible for oxygen binding. So after binding first oxygen molecule to hemoglobin, affinity of hemoglobin for oxygen increases. And second oxygen molecule binds more easily, and third and fourth oxygen molecules also bind more easily. So when oxygen is bound to hemoglobin molecules, this form or state of hemoglobin is called relaxed state or R state. So in this state, hemoglobin has high affinity for oxygen binding. So this state is favored in lungs, so oxygen can readily bind to hemoglobin. This cooperative binding of oxygen to hemoglobin results in sigmoid or S shape of oxygen dissociation curve. Oxygen dissociation curve tells us how partial pressure of oxygen correlates with oxygen saturation. In this graph, we have partial pressure of oxygen in millimeters of mercury on x-axis and oxygen saturation of hemoglobin on y-axis. We can see after some increase in oxygen saturation, there is steep increase in oxygen saturation with only small increase in partial pressure of oxygen. And this is due to increase in affinity of hemoglobin after binding to oxygen. However, this curve plateaus when most of oxygen binding sites of hemoglobin are occupied. And oxygen saturation cannot be further increased when hemoglobin is fully saturated with oxygen or when oxygen saturation is 100%. When we analyze this curve in reverse direction, we can see that initially, after significant decrease in partial pressure, there is no significant decrease in oxygen saturation. And this is due to high affinity of hemoglobin for oxygen. However, with further decrease in partial pressure, there is steep decrease in oxygen saturation. And this helps in offloading of oxygen and delivery of oxygen in tissues. This is normal oxygen dissociation curve. Suppose we have hemoglobin that has low affinity for oxygen. In this situation, oxygen will readily dissociate from hemoglobin and this will increase partial pressure of oxygen. So direction of reaction will be towards right. This will also shift oxygen dissociation curve towards right. In normal oxygen dissociation curve, when partial pressure of oxygen is around 25, hemoglobin is 50% saturated. However, if affinity of hemoglobin for oxygen is low, more oxygen will be released in solution and this will increase partial pressure of oxygen. In peripheral tissues, this helps in delivery of oxygen to tissues, as more oxygen is offloaded from hemoglobin and it becomes available for utilization by cells. This also means with a shift of curve to the right, a higher partial pressure of oxygen is required to achieve a given saturation. These changes happen in opposite direction when affinity of hemoglobin for oxygen is high. So when affinity of hemoglobin for oxygen is high, more oxygen will remain bound to hemoglobin and this will shift the direction of reaction towards left. And this will also shift oxygen dissociation curve to the left. We have seen how changes in affinity of hemoglobin for oxygen affects oxygen dissociation curve. We have a parameter that measures affinity of hemoglobin for oxygen, and this parameter is called P50. P50 is partial pressure of oxygen at which hemoglobin is 50% saturated with oxygen. In normal oxygen dissociation curve, when partial pressure of oxygen is around 27 mm of mercury, hemoglobin is 50% saturated. However, if affinity of hemoglobin for oxygen is low, more oxygen will be released in solution and this will increase P50. This means a higher partial pressure of oxygen will be required to maintain oxygen saturation at 50%. And if affinity of hemoglobin for oxygen is high, a lower partial pressure of oxygen will be required to maintain oxygen saturation at 50%, as oxygen will readily bind to hemoglobin. Now let's discuss which factors decrease affinity of hemoglobin for oxygen and shift the oxygen dissociation curve to the right. Earlier we discussed that hemoglobin has low affinity for oxygen in tissues. So which factors are present in tissues? We know in tissues we have high carbon dioxide levels, and carbon dioxide also leads to production of hydrogen ions. So we have high hydrogen ions and high carbon dioxide levels in tissues. And also if tissues are metabolically active, this can result in increase in temperature. So these factors decrease affinity of hemoglobin for oxygen, and this results in offloading of oxygen in tissues, and this also shifts oxygen dissociation curve to the right. Binding of hydrogen ion and carbon dioxide to hemoglobin actually favors 10 state or T state of hemoglobin, and we know that T state of hemoglobin has low affinity for oxygen binding. Opposite reactions occur in lungs. As carbon dioxide diffuses out, it reduces amount of carbon dioxide and hydrogen ions. And when hydrogen ion and carbon dioxide is low, it favors R state of hemoglobin. And we know R state of hemoglobin has higher affinity for oxygen binding, and it results in uptake of oxygen in lungs. Another factor that affects affinity of hemoglobin for oxygen is 2,3-BPG. In this graph, we can see how changes in affinity of hemoglobin affect oxygen saturation when partial pressure of oxygen is kept constant. So we can see that at same partial pressure, there is higher oxygen saturation when affinity of hemoglobin for oxygen is high, and low oxygen saturation when affinity of hemoglobin for oxygen is low. Now let's discuss how changes in hydrogen ion concentration and carbon dioxide affect affinity of hemoglobin for oxygen. In tissues, hydrogen ions are produced from carbon dioxide. And when hydrogen ions and carbon dioxide bind to hemoglobin molecule, it results in increase in strength of ionic interactions between adjacent globin chains. And when these ionic interactions are stronger, this favor T state of hemoglobin. And we know T state of hemoglobin has low affinity for oxygen bindings, and this helps in offloading of oxygen in tissues. This is called Bohr effect. So Bohr effect is decrease in affinity of hemoglobin for oxygen in presence of high hydrogen ion concentration and carbon dioxide. This helps in release of more oxygen from hemoglobin, and that helps in delivery of oxygen to tissues. We just discussed that increase in carbon dioxide and hydrogen ion decrease affinity of hemoglobin for oxygen. However, when partial pressure of oxygen is high, oxygen binds to hemoglobin molecules, and this favors the R state, and R state of hemoglobin has higher affinity for oxygen. However, R state of hemoglobin has low affinity for carbon dioxide, and this helps in release of carbon dioxide in lungs. Haldane effect is changes in affinity of hemoglobin for carbon dioxide due to changes in partial pressure of oxygen. So in lungs, when partial pressure of oxygen is high, it reduces affinity of hemoglobin for carbon dioxide, and carbon dioxide is released from hemoglobin and diffuses out of blood. Opposite changes occur in peripheral tissues, where partial pressure of oxygen is low. This increases affinity of hemoglobin for carbon dioxide, and carbon dioxide binds to hemoglobin